Hi there, this video outlines how to read codons and go through the process of transcription and translation using a codon chart. So I've kind of started to diagram here just a little bit of the double-stranded DNA helix. Now, when you're actually going to that flow of genetic information from DNA to RNA to proteins, you need to sort of kind of open up the gene area because it is, not only is it in a double helix conformation, but then it's often what we call supercoiled or twisted, so it's kind of inaccessible there. So I've started one half of the double-stranded helix, and then I'll go through and list the complementary base. And I'm just kind of doing this to remind you how to read those complementary bases. So in DNA, we have adenine, thymine, guanine, or cytosine. So adenine always pairs with thymine. And so when we see thymine, we're going to put in adenine there. Guanine is going to pair with cytosine, so adenine with thymine, guanine with cytosine. So I'm just going through and setting up my complementary strand. Now, when we look at the helical strand, at any given time, we can identify one strand as the coding strand and the other strand as the template strand. And this actually can vary based on the gene, but we have to just kind of pick a side here. So because I build in a complementary fashion when I go through transcription, I need to use the template strand. Actually, ultimately what I'm trying to get at is the coding strand in terms of the information that's there. But because we build in a complementary fashion, I'm going to need for the mRNA to build off of this strand. So in transcription, we're making mRNA. So here's our strand of mRNA. And now I can go through the template strand and build the complement to that. Now, all RNA uses uracil as its nitrogenous base instead of thymine. So when I go and I'm feeling like I want to write a thymine down to build the complement, I need to stop myself and remember that a uracil goes in that place. So if I see thymine, that still goes with adenine. And again, when I see adenine, instead of writing a T there, I would write a U there, the uracil. C pairs with G, C pairs with G, G pairs with C, T pairs with A pairs with U, A pairs with U, and so on and so forth. So remember that messenger RNA is a single strand. Now, if I want to go back and kind of check myself, I should notice that this mRNA looks an awful lot like that coding strand, because that's actually the code that I want. But because the enzymes build in a complementary fashion, you have to build off of the opposite strand. And the enzyme that's involved in this process, I don't have space for it over here because it would start over here, is called RNA polymerase. And a polymer, remember, is a long chain of any set of repeating units. So it's a very generic term. ACE is the enzyme ending, and RNA indicates what type of molecule I'm making. So I'm making a chain of RNA. This is extremely short. Normally for a protein, you would have hundreds or thousands of these bases here. So I'm just making a short one for illustration. Now, when we go to manufacture our proteins, the enzymes read in groups of 3, and that's what's going to specify our specific amino acids. So in DNA, those groups of 3 are actually called triplets. So I think that's a term people can be familiar with. In messenger RNA, we call those groups of 3 codons. So I've kind of artificially left a little space here. That's for myself. It's easier to read. Of course, in the real world, all these bases would be right up next to each other. But I just left a little space here for my eyes and for your eyes so we can kind of see how that particular group of 3 would work. Okay, so now remember that mRNA is going to go down to our ribosome. And ribosome has 2 subunits. That's kind of drawing some people jokingly call it the hamburger buns, kind of the top and the bottom piece there. And the mRNA is going to come down here and move through that ribosome. And then the transfer RNA is going to see what that, and this is a very sloppy drawing here of transfer RNA or tRNA, It's going to see what those codons are, and it reads them down here at what's called an anti-codon site. Kind of a small side fact, but anti-codon site. And then it's going to bring the matching or the correct amino acids going to come with it. So this is where the amino acid would go. And so there's different types of transfer RNA. It's still the same basic structure, but there's different variations on that read these specific groups of 3. Okay, so messenger RNA is down here. So let's write that really quickly again. And that's going to be read as, what we can do is go to a codon chart, and so we'll be read in those groups of 3. The codon chart is set up to read the mRNA, kind of saves you the step. You could write the anticodon out, which would be the opposite of this, would be the complementary sequence to that. But the chart is set up to kind of save you that step. Sometimes a wheel is used instead of a chart. So you go to that, whatever that is, that wheel, that chart, and you find that group of 3 and you read that. So the first one, hopefully, if I've done this correctly, should be a start sequence. And of course, there's not really a start in the protein, so I'm just going to put it in parentheses to remind you. So if you go to the chart, you should see the start. And these are going to be the amino acids that go in. So these little lines I'm drawing here are the kind of peptide bonds. And then GCA should be, and I'm going to double check here on my chart, but it should be alanine. And so you kind of have to practice this with a codon chart so you can see this happening. And then UUA should be leucine. And then if I've done this correctly, there ought to be a start here at the end, and that's this group here. And again, there's not really a stop, and there's not really a bond that's there. Now again, in the real world, this is going to be many, many amino acids long for a protein. I just made it short here to conserve space. So practice taking a DNA sequence, writing out the corresponding mRNA, and that's here. I've just copied it down. And then practice going to that chart or wheel and reading what those sequences should be. If you don't see there's a start at the beginning, chances are you accidentally used the coding strand, or perhaps you made a mistake in building the complement in the mRNA. So if you're not finding that start sequence, there may be a mistake in your steps. So go back and take a look at that. So that's all I have for the basics of codons, transcription, and translation. Just to quickly summarize, it starts at the DNA. It is the template strand that is used to make messenger RNA. The enzyme that's involved in that is RNA polymerase. And that messenger RNA travels over to the ribosome. Now in prokaryotes, because there's no nucleus, everything kind of gets mashed together and it can kind of all happen at the same time. But the mRNA has to bind to the ribosome, and then from there, those groups of 3 are going to be read by this transfer RNA, and it's going to bring over, let me just bring that over because we kind of made that. This would be a really, really short chain here. It'd be too long. Alanine and leucine. Again, it's going to normally be a lot longer than that. So it will read along there, and then when you hit the stop, then everything kind of dissociates and then the process ends. That's all I have for this particular area. Please contact me with questions. This is Corey Andres at Central New Mexico Community College using CNM Glass.